already. Let me start by a comment related to the previous talk. So I was once lecturing in Paris about our work. I presented data about anxiety testing. I did say that women came up with a higher tendency for anxiety in that test. And a woman got up in the audience and literally shouted at me. And she said, "This is the time and era that women should take their place in society, do important things. How can you get up and say that women are more anxious than men?" And I said, "Look, I couldn't agree with you more, or else I would not fly here to lecture. But we still need to say what the research came up with." So I'm at the interface between basic preclinical research with some collaborative studies that relate to human patients. And I'd like to touch upon the different aspects of this interface and perhaps tell you a little bit from my own experience about the neuroethical points that are relevant. So are there general neuroethical constraints to scientists? I would say that today brain research is a very powerful tool, which is what we just heard. And there might be possibly severe implications to subjects of brain research, which I will touch upon. And scientists naturally want their work to be known. They want to publish. In certain cases, perhaps publicity would not be advised. So that's the first ethical issue that I'd like to touch upon. So, for example, there might be cases when keeping silent provides an important advantage to the scientist society. And that goes outside of the brain research issue per se, but that's an important point. There might be cases where someone's rights might be deprived if findings are known, which is exactly the expression of those patients that Dr. Peel was, or volunteers that Dr. Peel was talking about. Perhaps the outcome of that test would change their life forever and deprive them of their rights. This is clearly an ethical concern. And there may be cases when you think, this is important, I want this to be developed. If I do that, then I need to write a patent first, because otherwise nobody will buy that project and develop it, and then society will miss the outcome, the benefits. That's another issue. And last but not least, you are thinking always, would publication of this finding empower unknown bodies that would risk subsequent damage? So this is clearly not limited to brain research. And indeed, you could think of scientists over decades and millennia that worked to embetter their societies, such as Archimedes, like Galileo, in our time, like Einstein. And I know, and we all read about Einstein's feelings that perhaps it would have been better if he didn't publish some of his findings. Others thought that this was very important, that it saved the world. So this ethical concern is not limited to brain research. Now the next point I mentioned is what happens when delaying publication can increase the prospects of utility, but that may involve ethical difficulties. So ever since the Human Genome Project, we keep thinking, should we run genetic screens for behavioral traits? And if we do, should that be publicized? Would a geneticist call the GP, as Dr. Peel was saying, and say, look, that kid is at a risk to become violent? Or shouldn't that be publicized? About patenting issues, should we write patents on brain intervening approaches? Those might be of benefit, but in other instances they might be really dangerous. Which control measures should be taken to avoid unethical steps by societies, by governments? So these are the very heavy topics that I will just touch upon, but I think we should all think about. So specifically for neuroscience, suppose we say if we publish anything, all the patients will be anonymous. No one will know who that is. Would that solve the ethical issue? And I'd like to argue that it wouldn't, because the published concepts may have wide effects. And I'd like to show you an example. And I regret not having kept the letter, but I was publishing work on exposure to insecticides and how that created effects that changed the behavior of subjects. And one day I received a letter on a letterhead that I've never seen before. This was several years ago. And the letterhead says, Her Majesty's Jail so-and-so. And the person who writes the letter is a convict, sits in jail for life because he murdered his in-laws, both of them. And he, apparently the jail has a superb library, he has internet connection, he read all of my papers, and he writes, I was stressed because I was forced to use insecticides in amounts that changed my behavior. And I'd like you to come and testify in my favor in court. And luckily I had the excuse that I'm not a real doctor, I'm only a scientist. But anyway, this is clearly an ethical issue. Okay. Or suppose we publish a protocol for control over someone's psychological responses, which is beyond the corner talking about changing by magnetic forces one's behavior. So could that publication empower unknown people or organizations with the risk of subsequent damage? Well, what comes, of course, to mind in that aspect is the Pavlovian work. It is not a coincidence that in the publication on Ivan Pavlov, we see soldiers behind. It is not a coincidence that the book about him talks about exploring the animal machine. This is impersonalized approach, which we know or we suspect has been used to control people in ways that we would consider unethical. So we do have a problem there. And I told Dr. Peel in the intermission that I would show something that I took out from Newsweek of this week, talking about ignoring the evidence, with a whole article saying that psychologists today ignore and reject science. They would read about the consequences of new methods, but they would not use them because they don't really trust science. And that article agrees with my personal experience. For example, I came up with a blood test for anxiety, and when I talk to psychiatrists, they say, why do you need that? If you want to know if someone is anxious, send them to the psychiatrist. They will talk to them and they'll come up with the answer. So there is also the ethical issue of different subdisciplines of scientists and clinicians that must talk to each other, even if that calls for translation of the messages and explanation in a language that subgroup of scientists is not used to. So another issue that comes up to mind readily, are there ethnically determined susceptibility genes for behavioral traits? I know that is being studied elsewhere, and some people would perhaps like eugenics to come back in this kind of disguise. And if you can find such behavioral traits, would you publicize them? If you found that someone is subject to phobias, should that be published or shouldn't it? My work is relatively well known because this is a very small country. So once I got this telephone call from television. They wanted to interview me because one of the people in the Israeli media is afraid of flying. So because I publish on anxiety, I should relate to that. I said, really, that's not my field, you know. I don't know much about that. But this is another question. And of course, we talk about societies, and that brings to mind the military. So this cover page comes from 1 year ago. It's about the U.S. military, just assuming that the Time editorial looks for validation of the data, they talk about 30% of the military troops of the United States in Iraq and Afghanistan as affected by pathological stress that brought them to clinical assistance. They talk about up to 17% of those soldiers under antidepressants. The number of prescriptions was unbelievable, but I really checked it. That's the number. Over 1 year alone in Iraq and Afghanistan alone, 147 million prescriptions. And worse yet, 40% of those army suicide victims, so those soldiers who commit suicide, were under treatment with these antidepressants. So I think that that is on the verge of a society using an unethical approach to solve a problem, definitely using inappropriate tools to solve it. So how bad is that? As this is my field, I keep looking for evidence. And again, compatible with what was said here before, I found a review saying that 13% of the United States adult population seek medical care because of anxiety and stress sometime in their lifetime. And this was before September 11. I'm sure it is higher now. The expenditure is immense, and half of that is spent by the patients themselves. And the patients are hospitalized six-fold higher in incidences compared to non-anxious patients, and are not hospitalized because of the anxiety. So this brings us to the main topic that I wanted to discuss, that is, brain research cannot stay separated from other fields of science and medicine. The brain talks to the body and vice versa, and they affect each other. And again, this calls for communication between subgroups of scientists that I'm not sure even read the same newspaper or journals or scientific professional journals. So our work relates to acetylcholine. And I wanted just to explain a little bit about it. And this neurotransmitter was discovered by an Austrian scientist, Otto Lowy. And it's about 100 years now from the discovery, so there's a meeting next month in Marburg. And what he did was he had a dream. He had a dream that said that there should be a chemical sent from a nerve cell to a tissue, not only electricity, as was believed at the time. And by the time he woke up in the morning, he forgot his dream. So he walked very frustrated all day. And when he had the same dream the next night, he got up, he got dressed, he went to the lab, he did something no Austrian respected scientist would have done at those times. He performed an experiment. So what Otto Lowy did was to electrify a frog's heart, dissected frog heart, which started pulsating, and then he took the medium in which that frog heart was sitting and which contained that chemical that he thought sent the message, and today we know this is acetylcholine. He put that medium on a second frog heart without electricity, and that frog's heart started pulsating. So he proved the concept, and he did get a Nobel Prize for that. He also put a plant extract on that frog's heart. And I want to tell you about that plant extract because it displays an unethical aspect of brain research. So the plant is called Physostigma venenosum. It is the origin of those cholinesterase inhibitors that today are used to treat Alzheimer's disease patients. And it was discovered by a British pharmacologist named Stanley in the 19th century in Africa as a witchcraft medicine. So this plant extract was given to someone who's tried for betraying the tribe, for example, or if a person suspected their wife that she was having an affair, they would bring her to the center of the tribe, force her to drink the extract of this plant. If she was telling the truth, she would throw up and survive. If she was lying, she would drop dead and solve the case there and then. Although we may argue whether this is an ethical approach, it certainly worked for many years. And the book on cholinesterases, from which I'm quoting, by Anne Silver, from a book from 1972 that I really like, tells about Stanley, who took this plant, it's called the calabar bean in Africa, with him on the boat trip back to England. And in his cell, he chewed on 1/8 of this bean. Nothing happened. So he chewed on 1/4 of the bean. And then he started having double vision, and he felt all those cholinergic effects that we know about. And the book says that he rapidly drank his shaving water and threw up, as those truthful ladies were doing, and survived, the book says, to perform yet more hair-raising experiments. So I would argue that this is an unethical approach to neuroscience, and today we wouldn't be able to do it that way. But anyway, Otto Lowy discovered the first neurotransmitter and the first protein that degrades that neurotransmitter. This is what physostigma does. It blocks acetylcholine esterase. And I had the naive thought about 25 years ago that this is a simple issue for study because there's one gene and one protein, I thought. And it's not a coincidence that we published a general review on the field only 2 years ago, and there's a lot that we don't understand yet, but this is my field of expertise. And he did win the Nobel Prize. Okay, so we cloned the human cholinesterase genes. And we could find that mutations in those genes, this is the first gene, the butyrylcholinesterase gene, it's located on chromosome number 3. And this is a family pedigree, an Israeli family with 2 parents that are heterozygotes, and their son is homozygous carrier to this mutation in butyrylcholinesterase. He was a soldier in the first Gulf War in 1991, and with all other soldiers, he had to take an anticholinesterase in anticipation of chemical warfare. And he got very depressed, insomniac, he lost weight. He got anti-depression drugs, which worsened his situation. And it's only when he stopped all medication that he got all better. So this is the atypical butyrylcholinesterase mutation, which is quite abundant in Israeli population. It's about 1 to 1,000. In Caucasian non-Jews, it would be 1 to 10,000. And I can tell you about a seminar that I was giving in a medical corps in Israel, where a general practically asked me, so how abundant is this problem in our soldiers? And I said, 1 to 1,000. And the general said, negligible. So nobody checks today whether or not a soldier should be allowed to take this medication, if and when the conditions come for that. The second gene is acetylcholinesterase. Here I can show you on chromosome 7 a carrier of a promoter mutation who is again subject to very massive stress susceptibility because of impaired expression of this gene. And the incidence now is 1 to 50 because this is a dominant effect. Still no one checks that either. So it came to the situation where one of my sons became, during his reserve service, an officer. He's what we call a Rav Seren, which would be what? Colonel? Major? Whatever. And he was part of a practice, a war game. And the person running the game said, so if things get to that level, you need to tell all the soldiers to take the medication. And my son said, Mom doesn't, my mom says no. So carriers of this mutation, when they are exposed to environmental levels of anticholinesterase, that is, they happen to live next to fields that are sprayed with insecticides, they suffer hyperactivation of the frontal lobe in multi-EEG analysis and downregulation of the deep nuclei associated with learning and memory impairment. Again, this is not checked for. The Ministry of Health in Israel checks sensitivity to anticholinesterases by running a blood test, and anyone who is below 25% of the average in the basic population would not be allowed to continue being employed with that exposure, but no one checks for environmental exposure of people who happen to live there. And I predict that with the coming years we'll learn more and more about such sensitive, fine-tuning susceptibilities, and someone will have to make the ethical decision to what extent to refer to these dangers. Okay, and I just wanted another anecdote to tell you so that this mutation was found together with that one in a cohort of people that immigrated to Israel from the Georgian mountains. So I had this PhD student, and I told him, look, we have a wonderful opportunity. A lot of studies were run on the biochemistry of cholinesterases in many different countries, except for beyond the Iron Curtain. And now we have a wonderful opportunity because so many of them immigrated to Israel. So we'll take a Russian population of immigrants and we'll find out to what extent they are sensitive. And he did the analysis and he comes to me very upset and he says, you told me you'll give me Russians and you gave me Iraqis. I said, why is that? Because the incidence is just 100%. It's the same. It can't be a population of Russians. And of course it was. So I sent him to be educated to the Museum of the Diaspora in Tel Aviv University, where he learned about the Georgian community, which, of course, was thrown from Jerusalem in 586 BC to Babylon. Today, this is Iraq, of course. And about 1,000 years later, part of that community walked practically to the Georgian mountains, where they stayed another 1,000 years, and then they immigrated back to Israel, complete with the genotype that they had in the Middle East. So there would be communities that keep and sustain their genotypic susceptibilities for thousands of years, and that's another issue that we need to keep in mind. Now, I'd like to touch about the communication between brain and body. So this is the acetylcholinesterase gene, and we know that its expression goes up under anxiety. And what we've checked in this study, which was run in collaboration with a Danish group, was the question, what happens under inflammation? That is, what would volunteers injected with an inflammatory marker react to in terms of cholinesterase expression? And what you see here is that the cholinesterase expression goes up. So that agrees with the genetic information, which is of no major consequence, but it also relates to anxiety. So think about this map as a map of anxiety, where the redder it is, the more anxious that person is compared to the baseline. This is the delta anxiety compared to the average in the population. And what you see here is the more acetylcholinesterase there is, the more anxious that person is. This is the blood test I was talking about. So now what we see is the cholinergic status, the capacity to break down that neurotransmitter, acetylcholine, is associated with inflammation because inflammatory proteins increase it. So it's associated both ways. And what we see is a vicious cycle between a neurotransmitter coming from the brain or the body and a reaction which is definitely bodily. And the question is, so how do we relate between these two subfields? And what I'm saying is brain researchers should definitely talk to clinicians, to inflammation specialists, to researchers from other subfields. Now, related to that blood test, I would like to share with you another communication that I got. So last year I was on sabbatical in the University of Berkeley in California, and I'm getting this email. And this email comes from a journalist of the ESPN, the sports channel, which I'm afraid I didn't know much about until then. So this guy tells me about a baseball pitcher. And that stopped me off the computer because I had to learn from other people in the lab, what does a pitcher do? So now I know. Okay, so this guy, his name is Dontrell Willis, and he's a pitcher with Detroit Giants. He's signed on a contract of $29 million 22 of which should be paid to him over the next 3 years, even if he doesn't play. And he came to the group and said, I had this blood test, and they told me I'm anxious, so I cannot play. So the journalist says, I looked for a blood test. I couldn't find anything but European AS paper from 2004. Is it commercially available? I said, no. We couldn't commercialize it because psychiatric specialists say, you know, you don't need a blood test for anxiety. So there is no blood test for anxiety. But this guy doesn't play. So by and large, he learned that I was in Berkeley, so we were discussing it, and now he plans to write a big article about anxiety in baseball. Apparently there was another pitcher who had, God forbid, to move from that job to the job of a catcher, which is less anxiety related. And I was talking by Skype to my son in Israel, here at the Hebrew University, and I was bragging about it. I said, oh, by the way, I was interviewed by a journalist from the ESPN. My son goes, Mom, ESPN is a sports channel. So, okay. So from this humorous issue, let me move to another one which relates to the ethical conduct of governments and organizations. So the Institute of Chemistry at the Hebrew University is named after Dr. Fritz Haber, who was developing nerve gases. And what you see here is the people who were interested in the findings of his research. What is less well known is that Fritz Haber was married to Dr. Clara Haber, who was the very first woman PhD of the University of Göttingen in Germany, and who did not accept what he was doing. And she tried to prevent him from pursuing this line of research, saying that this might cause a lot of damage to people. And he wouldn't, and she committed suicide. So here is the ethical aspect of chemistry, but as it relates to neuroscience. And of course what Fritz Haber did led on to the development of nerve gases, which are insecticides, if you wish, except that the target is not an insect. And it was used not only by vicious governments, such as at the Nazi regime, but also by terrorist movements. So in Japan in 1995, this same nerve gas was used and caused a lot of damage, much of which was never really reported because the Japanese patients are discouraged from coming to clinical assistance. Same agents are used by those governments who in the UN try to say that Israel is misbehaving, but use by themselves the benefits of this scientific approach. So there are clearly, and believe me, I'm not saying one should stop science from advancing. What I'm saying is we as scientists should be in contact with governments because governments should abide to ethical rules, definitely when it relates to neuroscience, but I believe in general. So to a much more pleasant topic, why write patents? So I came to this experience when I was receiving a grant award from the US military, and one of the conditions was whenever you reach this and that milestone, write a patent. And Ms. Slater, who sits here in the audience, knows that because she was involved with that from the administrative point of view. And actually, I like to quote Saint-Exupéry on that. And what he said in The Little Prince is, when you find a diamond that belongs to nobody, it is yours. If you discover an island that belongs to nobody, it is yours. When you get an idea before anyone else, you take out a patent of it. It is yours. And he continues, so with me, I own the stars because nobody else before me ever thought of owning them. So this was many years before lots on the moon were sold. But the idea is correct. If you don't write a patent, no one will like to develop your findings because companies are there to make a benefit. Okay, so I got this grant and I cloned the cholinesterase genes, and then my next aim was to create animals that would overexpress them. And this is the protein that protects from nerve gas. And I created these animals. And I'm quoting again a general from the US military. I was lecturing on my findings, and he said, okay, so you create the mice that overexpress this protein, and then we'll create the soldiers. He really did say that, except that when you create a mouse that overexpresses acetylcholinesterase, these mice are nervous. I mean, this is how they behave, and any of you who saw a mouse know that mice sit down and they scratch a little. These mice run. They run all the time. They always run to the right, and I don't think that reflects their political views. They are 15% lighter than little mates, which is nice, but if you put them to a swim test, they can swim like all mice. So this is a normal mouse, and the mouse first swims at the contour of the bucket to make sure they feel less threatened, and then they dare to go to the center. Now look at the engineered mouse. They never run to the center. They are afraid. And if you put them to a cereal maze, so this is, I hope you can see the mouse. We put it at one end, there's food at the other end. They know that very rapidly. When they get the food, we take them and put them again. Now here is the engineered mouse. And he goes back and forth and back and forth. And he forgot his glasses and he doesn't know where the keys are. I once said that, and someone in the audience said, just like me. So you would say, we know that when we are stressed we make errors of judgment. That's true. But at that point in time, I wrote a report, an official report to the American military saying you can either have a safe soldier or a wise one. So luckily they never engineered soldiers. So that brings us to post-traumatic stress. Walter Cannon used to say that stress reactions are fight or flight. We talk about stress responses as immense fear that would be related in many cases to war. This is Picasso's Guernica. Any of you who didn't see the recent animation movie about the Lebanon war, it's very highly recommended. And post-traumatic patients would suffer re-experiencing an arousal, an avoidance behavior, and they also suffer inflammation, but no one connected it because neuroscientists don't talk to inflammation scientists. And we know that acetylcholine blocks inflammation. So I would say that post-traumatic stress and inflammation are associated. And again, there is a vicious cycle that needs to be stopped. And I'd like to share with you, and this is just one little bit of real research, where we developed a DNA that blocks that overexpression that is caused under anxiety. And this is my PhD student, Gabi Zimmerman. And what he did was to stress mice. Now, how do you stress mice? This is totally ethical. You let them smell at the litter of a cat for 10 minutes. So we have another student in the lab who has a cat, and she brings the cat's litter. We need to sterilize it, or else they'll throw us out of the animal facility. But then the mice are really stressed, and we keep them for 7 days when they are either injected with the DNA blocking that expression or not. And then we check them for their jumping reactions or their daring capacity to go to an open space. And we keep remembering that acetylcholine blocks inflammation, so that our blockade of acetylcholinesterase should help the mice. And indeed, if we measure in the mouse blood, inflammation goes up when the mouse is stressed, and it doesn't go up when we block acetylcholinesterase. If we look at the brain, we see that the brain is hyperactivated under stress. This is 1 week after, but not in the mice that were injected with DNA. Now, there is a window of hope in PTSD, because when we have, God forbid, a terror attack, you keep hearing in the radio that there are that many wounded and that many panic patients. They would go to the hospital, and only about 10% of them would remain sick for life. So that would happen several weeks after, or maybe months after. So you have a window of time where prophylaxis can block that. The question is, would anyone be interested in developing something like that? We say today that there is a major problem in developing anti-malaria drugs because the patients are in unprivileged countries. They don't have the money to pay for that. So we went to pursue patients with post-traumatic stress. And we've done that, and we measured the inflammatory markers, some of which were really higher up in the blood of those patients. We then divided them to those with low and high markers, and we find that those with the higher markers were suffering more, experiencing more arousal, a little bit more avoidance. And finally, we ran the brain mapping, but this time definitely in a medical context. And we could find that those with a higher level of inflammation had a smaller volume of the hippocampus, which is known to be a parameter associated with post-traumatic stress. So all that with real results. But what about the governments? So we mention a lot books like 1984 when we talk about governments that enforce unethical rules. But I'm afraid it is not limited to 1984 to the 20th century. And I'd like to tell you a little bit of my experience or the way I look at the incidents when the Chechen rebels got over the theater in Moscow in 2002. These are the very few pictures that were taken on that were released because they were taken just by someone in the street. And look at that, what the soldiers did. So these are Russian soldiers, official representatives of a government. There were 800 people in the theater. The soldiers sprayed something. Then they got in and they took the people out. Obviously, the soldiers didn't wear masks. They were protected. Something protected the soldiers from what they sprayed. But about 1/4 of the people in that theater died. And now let's try to look at that. What really happened in Moscow? So if you look at the pictures, you see that the terrorists were deeply unconscious. There were no problems to take them out. What could it be? Now, I am involved in neurochemistry, so let's look at the neurochemistry of what could have happened there. Could it be laughing gas? No, it couldn't, because laughing gas goes under very rapid decay, and you would need very large volumes. This was a huge theater, so it could not be a laughing gas. What could it be? We anesthetize animals with halothane. Could it be halothane? No, it couldn't, because although it is still used as an anesthetic in Russia, the concentration in the atmosphere, again, is 0.8% in volume. Loss of consciousness does happen very quickly, but to a shallow extent, so people would not really be deeply unconscious. So this doesn't make sense. There are cholinergic blockers that would erase memory, but this induces psychotic states. And there is an international treaty saying you are not allowed to use that. But anyway, no one there seemed as if they developed psychosis. So this doesn't seem relevant either. This brings us to the incapacitating opioids. So you could use an incapacitating opioid, which would induce rapid and very deep unconsciousness and can be blocked by a blocker. And suffice it to say that the Russian government is not the only one who has used it, but to the best of my knowledge, they are the only ones who used it to such large numbers of people. And apparently they did not take care to treat those innocent theater goers, 1/4 of whom died. And if you look at the reaction to these agents, any pharmacologist would tell you, yes, sure, 20% would die. So, very last but not least, what about scientists? What about committees who decide about scientific findings? And I'd like to bring two names in that aspect. So I was delivering a lecture in San Francisco last year, and a woman approached me and said, "Did you hear about Rosalind Franklin? She was my aunt." I said, "Sure, I heard about Rosalind Franklin, but apparently her perception was that very few people would have." Rosalind Franklin was the person who created the data of refraction for DNA, for which Jim Watson, Maurice Wilkins, and Francis Crick won a Nobel Prize. She didn't. Many years later, Jim Watson wrote a book called The Double Helix, where he wrote about Rosalind Franklin. Rosie, he wrote, diminuting her, didn't look that bad. If she would have changed her glasses and did something with her hair, she would be rather nice. This I consider totally unethical. It's not the only country or subfield where that happens. So last summer I was honored by a Humboldt Prize named after Lise Meitner. Lise Meitner worked with Otto Hahn in Berlin in the Kaiser Wilhelm Institute on nuclear fission. Many years. She was the head of the Department of Physics in the Kaiser Wilhelm Institute in 1930, where very few women went to university. But in 1939, she had to run for her life from Germany to Sweden, where she continued to work, but she didn't have a lab. So she continued communicating with Otto Hahn. When the Nazi regime submitted his candidacy to Nobel Prize, they of course would not add her name. When he won the Nobel Prize, he wanted to share with her the money, because he felt bad. And Lise Meitner, and I'd like to end by going back to Einstein, donated all of that money to the Albert Einstein Fund for Peace. And I would think that although she wasn't a neuroscientist, her behavior was very ethical, and I'm really honored to have learned about her this last year. That's all I had to say, and I'd like to thank you.
Hermona, thank you very much for the exciting lecture. We have time for one question. If not, we will see after that in the panel. You are so wonderful. Nobody can do it better than you. Now
You're
I'm
exaggerating.
back on the chair.
Thank you. Okay. So if there are no more questions, okay, fine. So our next speaker is Professor Alan